Okay, thanks. Hi, everyone. My name's Michelle Przedborski. I'm a senior scientist at Certara. I'm going to talk about some of the work that I've been doing with them, but I didn't think to really prepare an introduction to discuss sort of like our, interaction, but I was a postdoc at the University of Waterloo prior to joining Applied Biomath, and then Applied Biomath was recently acquired by Certara. So the work I'm going to talk about today is called a quantitative systems pharmacology model to study the efficacy of therapies for the treatment of autoimmune disease. So there's been about more than 80 autoimmune diseases that have been identified to date, and it's estimated that about 5 to 8% of the world population is affected by autoimmune disease. There's a lot of interest lately in developing better therapeutics for these conditions. What I'm showing here are some models that have been published, QSP models, because we now know that QSP modeling is very important to help accelerate and de-risk drug development. And one thing that we noticed when we were getting into this space is that there isn't a whole lot of publications in regards to QSP models for lupus. And so we wanted to see if we could develop a framework to look at this disease specifically. So lupus, or SLE, is an autoimmune disease. It involves the recruitment and activation of various immune cell types, and it involves the creation of an inflammatory microenvironment, and ultimately will lead to dysfunctional B cells and the production of autoantibodies or autoreactive antibodies that ultimately will lead to damaging organs such as your skin, kidneys, and various other organs. Now, it's a very complicated disease, and what we wanted to do was see, given, oh, forgot about that. Yes, so there's a lot of interest in this space right now with developing drugs specifically for lupus. So as you can see, what I'm showing here are that you have anifrolumab and belimumab that were recently FDA approved. So with belimumab in particular, that was approved in 2011, and that was the first new drug for lupus that had been approved in 50 years. Since then, anifrolumab was approved, and now there's various clinical trials and that you can see are ongoing here for different drugs. Some of these drugs target CD20, for example. Some target CD40 ligand. And what we're going to focus on today are drugs that are targeting two targets simultaneously, which are BAFF and APRIL. And what we wanted to see is, given the complexity of lupus and all of the different cell types that are involved, is it possible that we can build a fit-for-purpose QSP model that focuses on specifically the B cell dynamics and the plasma cell dynamics and be able to recapitulate what's seen in clinic? So what we included in our model are several B cell subtypes, plasma cells, and then we also included the BAFF and APRIL, and these are known to modulate the survival of B cells. So for example, BAFF increases the survival for immature or naive B cells and transient B cells, and APRIL is involved in the maturation of B cells into plasma cells, secretion of immunoglobulins. And what I'm going to do next is talk a little bit about the model and how we calibrated it. I'll give some details, and then we'll talk about how we can use the model in a clinical setting. So this is just an overview of the model diagram. So what we have here, it's a three physiological compartments. So we have the central compartment, peripheral compartment, and then an immunological compartment. And we also included a second peripheral compartment just to capture the PK of the drug. So what happens is the drug is dosed to the central compartment, and then once it's dosed, either intravenously or subcutaneously, it can distribute to each of the peripheral compartments as well as the immunological compartment. When the drug enters the system, it's going to bind with BAFF, free BAFF, and APRIL in the system. And then when it binds to BAFF, what happens is you get a reduction in free BAFF levels in the system. And what we've done here is we've implemented the effect on the system to increase the death rate of the naive B cells. In terms of APRIL, when the drug binds to APRIL, you have a reduction in free APRIL levels in the system, and that's going to blunt the maturation of mature B cells into plasma cells, which are shown by these arrows here. And then we're also having an effect on the system where when the free APRIL levels are decreased because you're binding to the drug, you also have suppression of the secretion of immunoglobulins from the plasma cells. So in the model, we included intercompartmental transport of all of the soluble molecules, and we also have intercompartmental trafficking of the cells in each of the peripheral compartments. The key B cell dynamics are happening here in the immunological compartment. So we have the production of naive B cells, and then those subsequently mature into mature B cells, and then subsequently into either short-lived plasma cells or intermediate-lived plasma cells. So while all the B cell dynamics is happening here in the immunological compartment, we do allow the cells to die in the central and peripheral compartments as well. All in all, this model has 35 states or ODEs, 73 kinetic parameters, and then the key outputs for the model are the PK, immunoglobulin levels, and the B cell counts. So in general, the simulation strategy that we're using for this type of model is to start out by equilibrating the pharmacology model in the absence of treatment. So this means that you're going to have constant BAFF and APRIL levels, constant B cell and plasma cell levels, and immunoglobulin concentrations. And we use that steady state to initialize the treatment simulations. So when we apply the treatment to the system, we dose the drug to the central compartment, we're going to have the drug binding to BAFF and APRIL, and then that will cause the downstream effects by reducing the maturation rates of the B cells and plasma cells, as well as the production of the immunoglobulins. What we do then is we calculate the percentage change in naive and mature B cell counts and the percentage change in the serum IgA and IgG levels, and then we compare that with preclinical or clinical data. What we do typically is we start by building a preclinical in vivo model. And in this case, we had data, cyno data from the literature. So we start out by fitting the PK data, which I'm showing here. This was for single IV bolus and subcutaneous doses. And then we adjust the model structure if needed in order to capture the PK. Once we have that, then we'll start looking at fitting the PD. So in this case, we were looking at capturing the changes in mature B cells and IgG levels because those were available in the literature. One thing I want to point out is that there is a lot of fluctuations here in the placebo control for the mature B cells. So that's in the absence of treatment. And even for the other cases, after the dosing is stopped, which is indicated by this vertical line here, you can see that the recovery of the mature B cells is quite noisy, even for each of the doses that were looked at. So here we were really interested in looking to capture the overall timing and magnitude of the change in the mature B cells, as well as the immunoglobulin levels, which were much less noisy. Once we have our calibrated cyno model, then we want to do is build a human model. And this is accomplished by allometric scaling of compartment volumes, transport and elimination rates, and half-lives of the immunoglobulins. And then since we're building a model there where we want to capture the biology for SLE patients, we look to the literature to try and source some of the model parameter values based on clinical data. So that includes baseline BAFF and APRIL serum levels, baseline immunoglobulin levels, and cell counts in all the compartments. And then when we put those together, we end up with a preliminary human model. Then from there, we'll start to run simulations of the clinical dosing schedules, and we'll look at the model predictions of cell counts over time and immunoglobulin levels. And then we'll refine the model parameters so that we can end up with a calibrated human model. So when everything's said and done, this is what the calibration to the PK data looks like for clinical atacicept data. So you can see that the model can capture the dose response and does a fairly good job of capturing the single dose PK data. In terms of the multi-dose PK data, we can capture the troughs fairly well for that data. The next thing that we do is look at refining some of the PD parameters. And in this case, we're looking at applying doses weekly until day 22 in the simulations. So again, what we're trying to do here is capture the overall magnitude and timing of the decline in cell counts. You can see that just like in the cyno data, there is a lot of noise in the data. even in the case of placebo, as well as for the treatment cases. Overall, I think we do a fairly good job capturing the timing of the immunoglobulin reduction over time. I think if we were to do some additional model refinement and calibration, we could probably capture the recovery in the immunoglobulin levels a bit better. So the next thing we do is start to look at how our model's performing in different scenarios to see how, if it's meeting our expectations and our intuition, so that we have more confidence in the parameterization and calibration. So in this case, what we looked at was inhibiting just a single target, which is BAFF. And we accomplished this by keeping all of the parameters fixed except for the binding affinity for APRIL. We made the dissociation constant really large, so essentially it doesn't bind to APRIL. So I put these red X's here in the model diagram because these are the interactions that are going to be blunted in the pathway. And just given that we're only inhibiting a single target, we would now think that you would expect that the potency should be reduced. And when we run the simulations, you can see here, BAFF only simulations for both doses are shown here. And you can see that the reduction in immunoglobulins is lower than when you're inhibiting both targets. Another feature that we saw that was interesting is that there's a dose saturation that we observe. So above 10 milligrams per kilogram, you don't get any further reduction in IgG or naive B cell counts or the other clinical biomarkers. And this is similar to clinical data that's seen for belimumab, which is a BAFF-only inhibitor, which is shown in the slides on the slide on the right here. And importantly, the model was not calibrated to belimumab data. So this is sort of just an interesting feature that came out. The second thing that we did was we looked at another drug called povetacicept or Alpine 303. And in this case, what we did was we just updated the PK parameters based on what is reported in the literature for this drug. And as you can see, the model can capture the single dose PK data quite well. And when we look at the pharmacodynamic effects, that's what I'm showing on this slide here. You can see that the model can capture the overall trends in the dose response. There is an overprediction for IgG reduction. And in terms of IgA at the lower doses here, we are seeing that the model is certainly overpredicting slightly with the IgG changes. But I want to stress here that this is not calibrated to this data. And if we were to further refine these parameters, we could certainly capture this data better. So once you have a calibrated model, what can you do with it? I just want to spend a few minutes to talk about the model applications. So you can look at the, and this is certainly something that's interesting for people in drug development, is to look at how the drug parameters, such as the binding affinity, impact the efficacy of the drug. So what I'm doing here is applying doses weekly for a 100-day period, and I'm looking at the dose responses for IgA, IgG, mature B cells. Each of the different colors here corresponds to a different binding affinity for BAFF. And in the bottom row here, we're looking at changing the binding affinity for APRIL. This type of information is useful if you have a target efficacy in mind and you're wondering which dose you should start with to achieve that level of efficacy, or conversely, if you have a dose in mind and you're wondering what sort of binding affinity is required to achieve that efficacy. Another thing you can do is take a look at different dosing schedules. So you might look at changing the dosing frequency. So in this case, what we're doing is applying doses at different frequencies for a 100-day period. And we're showing the dose responses for IgA, IgG, and mature B cell reductions. Each of the different colors here corresponds to different dosing frequencies. So either weekly, bi-weekly, or once every 4 weeks. And as you can see, just as you might expect, if you're dosing the drug more frequently, then you can achieve a higher efficacy at lower doses. This type of information might be useful if you are looking, for example, in this case, at patient susceptibility to infection based on IgG reduction and how that might be impacted by the different dosing frequencies. As you can expect, there is certainly variability that's observed in patient markers in SLE patients. So in this case, we were looking at how variations in the BAFF and APRIL target levels might impact the efficacy. One thing that we found that was really interesting here is that even if we increase the target concentration by 10-fold in both cases, we could still achieve target coverage even at subclinical doses. And this you can see based on there not being a whole lot of variation in the IgA or IgG levels over time between these different cases here. So what we found is that there are going to be differences that lead to different patient responses, but BAFF and APRIL levels are not going to be primary drivers of those differences for atacicept. And then the last application I'm going to talk about is just the global sensitivity analysis you can do. So in this case, what we're doing is I'm plotting here the partial rank correlation coefficients for the top 10 most sensitive parameters for IgG reduction in the system. And here I'm showing in the bottom plot the IgG reduction over time for 100 randomly chosen parameter sets. We sampled the parameters from uniform distributions and physiological ranges. And what you're seeing here are, given all these sensitivities, these represent potential areas for therapeutic optimization. Another thing that was interesting is that we saw that the parameters that show sensitivity and the magnitude of those sensitivities are distinct for IgA and IgG. Okay. So that's basically the end of my talk. I just wanted to give a quick summary. We developed a fit-for-purpose QSP model for B-cell targeted therapeutics in lupus, and we calibrated that to cyno and human data for atacicept. And the model qualitatively captures trends for both BAFF and APRIL blockade. There's several things we could do to expand this model, including incorporating additional in vitro drug binding and efficacy data. We could consider higher order binding of drug to BAFF multimers or heteromers, as well as incorporate other cell types into the system, such as T cells and dendritic cells and the feedback loops between them, as well as cytokines that are relevant to SLE pathophysiology. Another interesting extension of the work would be to calibrate this to other molecules like telitacicept so that we can have more of a platform-based type model. I also wanted to give thanks to my colleagues who were involved in the development and initial calibration of this work, Sahili, Sheetal, and Faye. I'll take your questions now.